Hello, welcome to 13 Universal Learning Beyond Barriers, and this is A-level CIE Biology with Shedro. I'm Shedro. So we're going to continue with All Topics Series 7. Okay, so come with me. Let's dive in. So we're going to be doing some topics that we didn't do in other series 6, 5, 4, 3, 2, 1. Okay, so come on. It's time to apply some concepts. So let's go, guys. Come with me. So the transmission electron micrograph shows a cell in a stage of mitotic cell. Come on. This is beautiful. Isn't this beautiful, man? Come on. It's beautiful. All right, now let's go. So which statement explains why it is difficult? Why is it difficult to identify the stage of the mitotic cell cycle shown? Why is it difficult? All right, these are the options. So I want you to work it out. Okay, pause this video and test yourself. Okay, pause it and test yourself. All right, now you are back. They said which statement explains why it is difficult to identify the stage of mitotic cell cycle shown. So let's go to option A. Chromosomes have supercoiled and are visible, but centrols are not visible. Now we don't need central to identify it here. And again, this is not talking because where the supercoil, this where the supercoil is in prophase. Prophase is when chromosomes or chromatins coil up or supercoil or condense, right? This is not prophase. All right, we can see it clearly. This they have separated. It's not prophase, so it's not why is difficult to identify. All right, so B says anaphase may be continuing or telophase may be start. That's why this is why it is difficult, because it appears like anaphase. Why is it like anaphase? All right, they have separated. Can you see? The sister chromatids have separated at the poles. So we don't know whether anaphase is continuing, okay, or telophase may be starting. Now telophase may be starting. We don't know. We're not sure. Okay, of course, when telophase starts, begins, you're going to see my nuclear envelope reforms, and of course, nucleolus reappears and all of that. Okay, so this is where the problem is. But let's check the other ones. See, says it is unclear whether the electron micrograph shows two cells in metaphase. So this is not metaphase. That's not why. If it were metaphase, they will line up at the equator. Can you see? They will line up here. That's a typical characteristic of metaphase. But here they are separated. All right. So this says some people may consider the interphase to have. So no, this is not interphase because they have separated. Can you see? Interphase, these are not visible. Chromosomes are not visible, right? So the answer is B. Okay, anaphase may continue or telophase may be starting. All right, let's go to the next question. Come on. During lag in strand replication, short fragments of DNA are produced. Okay, the fragments are joined together by DNA ligase. Right, which role correctly describes the structure of these short fragments of DNA? Yeah, that's it. So guys, pause this video and solve it by yourself. Do it by yourself, okay? Do it. I'm not going to do it for you now. Do it. Pause it. Okay? Apply the concept. All right, don't forget that you have to understand the concept and you have to remember this topic. You can't apply concept you do not remember. And the most important skill in biology is memory. You must remember exactly the concept that you understood, that you have learned. So do not just study to know, understand. You must learn to remember. Okay, is a key, very key skill in biology exams. You must remember stuff. Okay, let's dive into it, guys. Come on, let's do it. Now remember that during DNA replication, you see, if this were to be DNA, all right, molecule, double helix, can you see it? Wind it up. Now this double, because two strands. Okay, so it's time for this guy to replicate. Remember that the first thing that happens is that is gonna, okay, unwind. DNA helicase will cause it to unwind, breaking the hydrogen bonds between the complementary bases on opposites on the two strands. And then we have something like replication effect like this. Can you see? Yeah, so replication effect, it's formed like this. Okay, so it's unwinding, it's unzipping process. Okay, yeah, can you see that? So it's still unwinding. Guys, don't forget that, this is direction of unwinding process. Okay, so that is unwinding process or unzipping process. So that's direction, the direction. Okay, now the strand that is always, all right, in the same direction, that is copied in the same direction, all right, with the direction of the winding process is 3' to 5'. Okay, all right, it's always on top. Okay, now the other strand here, which is 5' to 3', you can see this 5' to 3'. How is it copied? So replication here is discontinuous because the copying process, all right, is in opposite direction to the unwinding process. Okay, so you see that when this is being copied, we're going to produce another strand here. This is the strand. This strand is produced using we're using this, of course, remember, we're using this as a template. Both of them are used as template. This 3' to 5' template is going to be copied from the template, all right, from the 3' end. DNA polymerase always copy from 3' end. That's why it's starting from here, all right. But you see, as this unwind, it copies it. So this strand that is formed here is the leading strand. Okay, that is the leading strand. Can you see the leading strand? So I will call it leading strand LD. That is the strand that is formed there. We're not going deep into this. Okay, now remember that this strand is copying in a discontinuous, all right, fashion, because it copies in the opposite direction as the unwinding direction. Is that okay? So it has to wait for a section to unwind and then it copies it, and then wait for another one to unwind, it copies it. All right, so it copies different sections, right, as the DNA double helix unwind, and that leaves us with fragments. That's what I'm trying to say here. So that leaves us with fragments of DNA. So these fragments of DNA, that short fragment, is what we call Okazaki fragment, guys. That's Okazaki fragment. So Okazaki fragment will later be joined by DNA ligase, right? DNA ligase forms the sugar phosphate backbone, fill up the gaps, all right, to form the lagging strand. So this strand that is formed here, all right, using Okazaki fragment, is the lagging strand. So I'm call it LG. So that's the strand that is called lagging strand, right? So the other one up there, okay, that involves continuous replication, okay, we're using the 3' to 5' strand to form its leading strand. So this is going to be lagging strand. Okay, so watch this, guys. I want to show you something. So just before we continue this question, these are preambles you have to understand. So if, for example, we have base A here, all right, okay, oh, this will be T. Okay, let me try to adjust it. Okay, don't worry. Okay, let's assume we have at this point we have C here. Okay, this will be G. Okay, because they are complementary. Okay, all right, and here, if you have C there, this also will be G because the template strands are complementary. This will be G, and this will be C. Okay, this will be C. Do you get? Yeah, you get that. Okay, so if this is C, this will be G. Okay, this leading strand, okay, has the same base sequence as the template strand here, 5' to 3'. Okay, while the lagging strand has the same base sequence as the template strand there. All right, that means the leading strand and the lagging strand, they will have complementary bases. Okay, let's go back to the question. Which row correctly describes the structure of this short fragment of DNA? Okay, this short fragment of DNA, which is the Okazaki fragment that is used to make lagging strand here. Okay, now they say how base sequence that are complementary to the newly synthesized leading strand. Yes. Okay, so whatever base that is here is complementary to the leading strand. Do you understand? Because these two template strands are complementary, so the newly synthesized strands will be complementary too. Okay, so that means that A and B are eliminated because the base sequence in leading strand and lagging strand must be complementary. So we eliminate A and B, so we focus on C and D. So let's go to the next one. It says consists of one polynucleotide strand. Yes, the ligand, because they're asking us which role correctly describes the structure of this short fragment of DNA. Now, okay, consists of polynucleotide strand. Yes, the short fragment, when they are joined, it forms the ligand strand, and it's just a strand. It's a strand. Okay, so it's a polynucleotide strand. So it's yes between C and D. So we go for D because this means yes. Contains only carbon, hydrogen, oxygen, nitrogen. Guys, don't forget that DNA generally, okay, the elements that make up DNA, all right, we call it chomp. Chomp is CH, okay, that's carbon, hydrogen, oxygen is there, nitrogen is there, but there's an important one there, which is phosphorus. Okay, so DNA or nucleic acids, they all have these elements: carbon, hydrogen, oxygen, nitrogen, phosphorus, but there's no phosphorus here, so this is wrong. Okay, so that's why D is the answer. Pause this video and check it again. All right, so the answer is, of course, this key is correct, wrong, okay, is D. That's D. Let's go on. All right, the genome of bacterium Escherichia coli has been altered to enable it to code for an amino acid that is not found in nature. Okay, all the ATC DNA stop triplet on the strand of DNA that is transcribed have been substituted to ATT. Okay, all right, that's ATC substituted from ATT. So, all right, so C substituted with T. They say the ATC triplet can then be inserted to code for the new amino acid. A new transfer RNA can then be constructed to carry out the new amino acid. Okay, so what is the anticodon of this new transfer RNA? Okay, so I want you to pause the video and try it. Okay, remember that the new one is ATC. This ATC triplet can then be inserted. So this ATC is on the base triplet of DNA. Okay, what we're looking for is the transfer RNA anticodon. So the DNA itself, that's the base triplet, okay, is ATC. Okay, so ATC. Now in our video on nucleic acid, I spent time to explain that. So if you have the base triplet on DNA ATC, this ATC, sorry, this is ATC, and then what would that be in transfer RNA? What would be the transfer RNA anticodon for that? Sorry for the write-up, okay? What would it be? So I told you that the formula is that the anticodon of transfer RNA, please watch this, don't forget it. You see, the anticodon of transfer RNA, remember, and the base triplet of DNA, they are both complementary to the codon on mRNA. That suggests that the anticodon on tRNA should be similar to that of base triplet of DNA. If both of them are complementary to mRNA codon, then they should be similar. The only thing you do is that whenever you see T, you put U. So we have ATC, see, we have ATC on triplet of DNA. So what would that be for tRNA? I will just remove the T there, put U, because RNA doesn't have thymine, they have uracil. Instead of being ATC, so here it will be AUC. I just removed the T, put U. AUC is the anticodon on tRNA. Okay, so comment, let us know what you're thinking. Now look at this: 40% of bases in a section of a non-transcribed strand of DNA are purine molecules. 40% of bases are purine molecules, non-transcribed strand. Okay, what would be the total percentage of cytosine and uracil bases in the primary transcript that is transcribed from the other strand of the DNA? All right, so what if you get this answer, let me know. I'll buy chocolate. Okay, I'll buy chocolate. Try it. I'll buy chocolate. All right, so let's pause it and work it. All right, so watch this, guys. Remember that DNA has two strands, right? Let me use it like this. These are two strands of DNA. All right, Of course, one of them is going to be used as this is non-transcribed strand. Okay, all right. One of them is the transcribed strand. Is that okay? All right. During transcription, we use only one strand. Just a section of that strand will be copied. All right, to make mRNA, which will be used to make DNA. Is that okay? All right. Now, so they're asking us. They say that 40%. If you check here, they say 40% of bases in a section of a non-transcribed strand of DNA are purine molecules. This is non-transcribed. 40% of all the bases here are purine. Purines are double ring, all right, bases, double ring structure bases. Okay, so if 40% on the non-transcribed strand here, watch, this is 40%. All right, 40% are purines. Please let me use Pu for purines. Okay, are purines, right? Yeah, this, oh sorry, that's Pu. Okay, purines, right? 40%, all right, of the bases are purines. All right, now that means that 40%, now 40% are purine on that strand. Okay, that means that 60% will be pyrimidines. Yeah, that balances it. So 40 plus 60 is 100, right? So, okay, they're always purines and pyrimidines. So pyrimidines I will represent using PY. Okay? All right. So, so if you have 40%, all right, purine on this strand, this 40% purine will bond with, all right, 40% pyrimidine. You remember? All right, because purines form hydrogen bonds with pyrimidines. Adenine is a purine. It forms two hydrogen bonds with thymine, which is a pyrimidine. Guanine is a purine. It forms three hydrogen bonds with cytosine, which is a pyrimidine. So if I have 40% of purine here, then 40% of pyrimidine will be here. Okay? Are you following me? Let's crack this question. Let's wear this question out. So that means 40% of pyrimidines here, okay, will be, of course, 40% will be pyrimidines here. Pyrimidines, remember our, okay, symbol is py for pyrimidines. Now if I have 40% of pyrimidines there, all right, that means 60% pyrimidines in this first strand will give me 60% of purines, because purines bind with pyrimidines. All right, so I'm going to have 60%, sorry. So watch me please carefully. 60% here, okay. Now, okay, of purine, Pu, yeah, purines in the transcribed strand. Okay, so we're not doing bad. So remember, 40% of bases in section of a non-transcribed strand, that is here, are purine molecules. What will be the total percentage of cytosine? Remember that cytosine and uracil are pyrimidines found in RNA. Okay, basis in the primary transcript that is transcribed from the other strand. The other strand is this one. This is the template strand. Okay, this strand is going to be used to make messenger RNA. Okay, so messenger RNA has cytosine and uracil as the pyrimidines. Okay, so they're now asking us for, what are they asking us? They're asking us for the percentage of this cytosine and uracil in the messenger RNA, this one. Okay, that is the messenger RNA. Yes, so let me just say mRNA. Okay, yeah, mRNA. Yep. So remember, so mRNA. So remember guys, that these two are pyrimidines, right? So pyrimid, and this is going to be used as a strand. The pyrimidines here, all right, will have to pair with the purines on this strand. How many purines do we have? 60. So 60 purine here will give us 60% pyrimidines, because cytosine and uracil are pyrimidines. So this is a template strand. So I'm going to copy 60% of it into here. I mean, this is pyrimidine. So during transcription, remember that purines, the RNA nucleotide bases pair up with, the bases on the template strand. So 60% here, purine will give me 60% pyrimidine. Okay, so 60% pyrimidine will be here, and the pyrimidines are two of them, cytosine and uracil. So this is 60%, okay, of pyrimidine, which are represented, right, by cytosine and uracil. Okay, they are the pyrimidines. So the answer to the question is 60%, right? So 60% of the primary, because this is the primary transcript, okay? After transcription, this is the first one from the primary transcript, which will be converted to mRNA by splicing, all right? So, yeah, so 60% pyrimidine, purine here will give us 60%, right, pyrimidine there, okay? So watch this video again. If you have a question, ask us. So the answer is 60. Let's go on. D. All right. Which statement about xylem vessel element is correct? Now, try this. Which statement is correct about xylem vessel element? Is correct. A says, pause the video and try it. Hollow vessels enable the constant movement of water up and down. Water only moves up in xylem from soil right to the leaf. It doesn't move down. So this is wrong. B said pits enable the movement of water into adjacent cells. Yeah, pit is the unlignified part of the xylem, so it allows lateral movement, okay, movement, all right, between two xylem, okay, or to neighboring cells, okay. So that seems correct. So if this is xylem, this is another xylem, okay, because pit is unlignified part, so water can move laterally from this one to this one, okay, all right, so it can move laterally. Okay, so that B looks that is correct. But let's check the vessels contain numerous mitochondria to generate ATP. Now xylem vessels don't have organelles, they are empty, hollow. All right. So vessel contains perforated cross-link C plate. C plate is not found in xylem vessel. They do not have any end walls. This is for phlegm sieve, phlegm sieve tube, right? Phlegm, okay. All right, so it's B is the answer. Right, let's go to the next question. Ammonium ion can enter the xylem of plant roots by two pathways. In the apoplast pathway, ammonium ions move through cell wall. So apoplast is through cell wall, okay, and that this pathway is blocked by the Casparian strip, which is a band of suberin, okay, hydrophobic. They don't allow charged molecules to pass. Ammonium ions then enter the cytoplasms of root cells. All right, so in the simplest pathway, ammonium ions move through the cytoplasm, okay, of root cells. That's through plasmodious matter. A scientist measured the uptake of ammonium ions into the xylem of Arabidopsis thaliana. Okay, the SGN3 mutant of A. thaliana does not have Casparian strip. Okay, in the SAG3 mutant, ammonia ions can enter the xylem without entering the cytoplasm of root cells. All right, of course, they have removed the Casparian strip, so Casparian strip prevent apoplast, but then now they removed this, so it can actually move through the so that's what they're trying to say, that in this one doesn't have Casparian strip. So they say in these ammonia ions enter the xylem without entering the cytoplasm, so they can follow cell wall, right? The bar chart shows the scientist. Okay, look at it. Which conclusion is correct? Which one is correct? This is testing your ability to analyze, analytical thinking. Which conclusion is correct here, guys? Let's follow it. Remember, this is low concentration of ammonium ion. This is high concentration of ammonium ion, right? So this which conclusion is correct? Fewer ammonium ions enter the xylem when they have to move through the cytoplasm of root cells. Okay. Now, fewer ammonium ions enter the xylem when they have to move through the cytoplasm. Now, moving through the cytoplasm of root cells, when they have to move, they say fewer ammonium ions enter the xylem when they have to move through the cytoplasm of root cells. Okay, so this is not a conclusion shown here. Okay, why? because they're telling us that fewer, right? Now, they say fewer enter. Now look at these ones here. This is the non-mutant. So this one has Casparian strip, all right? This has Casparian strip, and it's telling us that the fewer will enter when they have to move through. So these ones here, the non-mutant, all right? Ammonium ions cannot pass through the cell wall because of Casparian strip. So they have to move through the cytoplasm. Can you see? But they are higher than the ones that don't have Casparian strip. So it's not fewer. So for both low concentration and high concentration, the ones that have to move through the cytoplasm, that's the one that are non-mutant, the ones that have Casparian strip, they have higher, can you see, higher uptake of ammonium, all right, ion. So this A is wrong. Doesn't, okay, describe it well. B says more ammonium ions enter the xylem at low soil concentration of ammonium ions. So if you go to low soil concentration, okay, they say more ammonium ions enter. If you compare low and high, high is that more ammonium ions enter at high compared to low. So that's wrong. High has higher uptake, okay, for both non-mutant and SGN mutant. All right, so B is wrong. C says the Casparian strip acts as a barrier to reduce the movement of ammonium ions into the xylem. This is wrong. If it acts as a barrier, that means that the mutant should have higher uptake, all right, because Casparian strip has been removed here. Why is it not higher than the one that has Casparian strip? Why? Okay, look at it. In both low concentration and high concentration, all right, the one that has Casparian strip has higher uptake. So C is wrong. So D is our answer. Let's look at it. The loss of Casparian strip has little effect. Yes, little effect on the movement of ammonium ions into the xylem. So the loss of it. So this SGN3, all right, has no Casparian strip. So both in high and low, all right, it has no effect. Do you understand? Because they're almost the same, all right? And of course, the one that has Casparian strip has higher uptake a bit, so it has no effect. That's what the scientists discovered, this analytical skill, trying to analyze data, find out trends and interpretation, explanation, reason for that trend, okay? So the answer should be D, guys. That's D. All right, come on. This is taking time. Let's go. Now, which row correctly shows processes required for the movement of water from a root hair cell to the atmosphere? Yeah, which one? So cohesion requires it, right? Why does it require it? Somebody's asking that. I know you're asking about that. Now, cohesion is required because for water to move up xylem, remember, water column has to be set up. Oh, what am I drawing? Pardon my drawing. Okay, this is xylem. So this sphere is water. So water column has to be set up. So water column has to be set up. And the only thing that can help to make water column is hydrogen bond between them. That's cohesion. Cohesion. All right, cohesion is required. Yes, diffusion is required. Water vapor diffuses from the air space in the leaf through the stomata to the atmosphere. So diffusion is required. Evaporation is required. Why? Because water molecules evaporate from the cell wall of mesophyll into the air space of the leaf. Okay, so all three of them are required. So the answer is A. All three of them are required. Which process can be carried out by a mature red blood cell? Okay, active transport. Yeah, most likely. Cell division. Mature red blood cell has lost the nucleus. All right. Mature red blood cell doesn't have nucleus, so that to create enough space for more hemoglobin. All right, so it cannot divide. Okay, I've given you a clue. What's the answer? Okay, pause it and check it. So it cannot carry out cell division. It cannot carry phagocytosis because it's not a phagocyte. Red blood cells are not phagocyte. Protein synthesis is not possible because protein synthesis requires transcription, and mature red blood cell does not have nucleus. So how will transcription occur? So protein cannot be synthesized. So it's only active transport that is possible. That's A. All right. The photomicrograph shows 3 white blood cells labeled XYZ. So what is the answer? Let's check what it's asking us. Which row correctly identifies these cells? Which row correctly identifies? So if you see this one, now see, anytime you see, this is nucleus, these 10 parts are nucleus, okay? Because of chromatin inside of them, right? So the one that has compact nucleus that covers more than 2/3 of the cytoplasm is always lymphocyte. So X, where is lymphocyte? A and B. So eliminate C and D. The one that has lobed nucleus. Lobed nucleus is neutrophils. It allows neutrophils to squeeze through the endothelial pores of capillary into tissues when they're being invaded. Okay, when tissues are invaded, so neutrophils can squeeze through it. So it has lobed or irregular nucleus. Okay, go to Y. So remember we're on A and B. The one that has neutrophils is B. So B is the answer. So the one that has kidney-shaped nucleus, can you see, is always monocyte or macrophage. All right. So the answer is B, guys. All right. The last question there. When a blood vessel is damaged, it can result in problems in the body. Which role identifies the blood vessel that could have been damaged? Yep. So it's a blood is not being distributed to the organs that need it. Which vessel distributes blood to organs? Artery. Artery takes blood to the organs, right? Which of them is closest? Now, if you come to there are two types of arteries. We have elastic artery, muscular arteries. So the one that is closer to the tissues is the muscular ones. Okay. So we eliminate A and B. Muscular arteries are the ones that are close to the organs, destinations, right? And they carry blood pressure. Now, there's a blood is not returning to the heart and is collecting in organs. So which blood vessel returns blood to the heart? Vein. See? Okay, between these two, vein. So the answer is okay. Blood surges mean that relative constant blood pressure is not maintained. Okay, so that means that which vessel carries blood at high pressure is artery. All right, which one is actually elastic artery? That's aorta. Okay, aorta is closest to the heart. It carries blood at high pressure, so it has a lot of elastic fiber. So it's an elastic artery. So the answer is delta. So thank you guys for staying with us. Please share these videos, comment, like, and then of course share with your friends. Thank you. This is Shedro. See you in next series. Bye.